Good morning, everyone, and welcome to our last webinar for the year. We appreciate your interest and joining us today. I have the pleasure of introducing Dr. Ruth Petran. Dr. Petran is the Vice President of Food Safety and Public Health at Ecolab. She provides technical expertise and consultation to internal and external customers on food safety and public health issues by identifying and tracking emerging food safety trends and new control strategies. Prior to joining Ecolab, Dr. Petran worked in microbiology and supplier quality at various manufacturing organizations and led food safety assessments at food manufacturing facilities worldwide. Dr. Petran is a member of many prestigious food safety organizations and committees, and she's advised the Minnesota Department of Health and Agriculture on needed revisions to the state's food code. Dr. Petran has worked on development of supplier auditing programs and is a recognized HACCP instructor in Ecolab's advanced HACCP course, which is accredited by the International HACCP Alliance. Dr. Petran has a Bachelor of Science degree in Consumer Food Science from Cornell University, a Master of Science degree in Food Science from the University of Minnesota, and a doctorate in Public Health from the University of Minnesota. Her thesis focused on the value of leveraging data from health department inspections to improve food safety. With that, I'd like to introduce Dr. Ruth Petran, and again, thank you for listening today. Thank you.
Thank you, Miriam, and thanks to you all for joining us today. We really appreciate your participation. So the focus of our webinar today is talking about a new detective that we have in fighting foodborne illness that can help us relate foodborne illness incidents across the country and actually across the world to better get at this thorny food safety problem. So today, we will talk about how food safety seems to be even much hotter news than previously. Some of this is because of enhanced detection technologies, which can certainly provide us public health benefits, but industry needs to be ready to react to these improved capabilities because these implications from such detection technologies require prompt actions to manage the risks that are identified. So if we think about the picture of foodborne illness today, frankly, it's not pretty. Each year, it's estimated that 600 million people get sick because of foodborne illness around the world, and maybe 420,000 people die. In the developed world, this translates to about 1 in 6 to 8 of us getting sick each year. Clearly, these numbers are higher than they should be. The most recent data came to us from the World Health Organization, who undertook a program to estimate the global burden of diseases from a number of foodborne illness agents. What they found is that the African region has the highest disease burden, followed by Southeast Asia, but there is some global variability, yet remarkable similarity in what the actual causes of illness are. Generally, in the developed world, illness is attributed to norovirus, Campylobacter E. coli, and Salmonella predominated, and the developing world saw issues attributed to these agents as well, but also expanded to include hazards from chemicals. We know that Salmonella, both the typhoidal form of Salmonella and non-typhoidal, which is typically associated with foodborne illness, was responsible for the most deaths, followed by E. coli and norovirus. And it's important to keep in mind that 40% of those who were suffering were young children. And this slide details some of these data. And this is the U.S., or U.S. is included within this region. What the World Health Organization did is divide the world basically into a number of subregions, and this is where the U.S. fall. You can see that the general burden of illness, of foodborne illness, is the lowest in America, also in various European countries here, and Australia and New Zealand appear here. But the rest of the world, including Africa, various places in the Middle East, and then Southeast Asia, and more Asian countries here, are quite a bit higher. And they were able to break down actually what caused these illnesses. So this gives us some clue as to what the actual agents might be that causes illness. But clearly, it's a big problem. Thinking about just the U.S. for a minute, and I do think these data would be somewhat reflective of the rest of the world to some extent. We know that norovirus is the leading cause of foodborne illness here. And we did look at these data to see what might be causing this increase, which is this dark blue line here. This is norovirus. There were enhanced methods, which relate to our topic today, for better detecting norovirus, put in place around the late 90s. And this did lead to an increase in reporting of illnesses, but I think is also reflective of the fact that our technology got better. Because back here, this dark gray-black line represents unknown caused illness. So we didn't know what caused these illnesses back here. The suspicion is likely they were norovirus, but we just didn't have the technology in place to detect them. Once we had that in place, you can see how the number of unknown cause illnesses dropped and norovirus went up, but at least this gave us some clues about where and what the most significant issues were. So what foods should we be concerned about? These are data from the CDC indicating a number of various food commodity groups that have been implicated in illnesses. And we see illnesses attributed to what we would customarily think about, protein products and produce and so on. Fish also, and generally that's due to issues with temperature control in the catching of the fish. But this can help us hone in on where do we need to focus efforts. Also, thinking about what locations are the riskiest. And these data are from the CDC as well, indicating that in general, food service environments were where people reported their illnesses occurring at most of all. And although 65%, for example, attributed to restaurants is certainly high, we even need to keep in mind these lower percents. For example, 1% occurring in schools. Remember, the U.S. estimated illness numbers are up at 48 million each year. And clearly 1% of that would be close to 5 million illnesses occurring among schools across the U.S. So that's a big number, and again, we can't ignore even these small percentages within this breakdown. But where do we focus efforts, and what behaviors should we focus on? Well, CDC gives us more clues about this when they describe the contributing factors to foodborne illness based on reported data. And we have bucketed these into a few different areas related to issues with temperature, including improper holding or inadequate cooking, issues with people and their personal hygiene related to things like hand washing, working when sick, also contamination from the equipment or environment, and then unsafe source or the food supplies as well. But layered over all these basic facts that we know about foodborne illness are several realities that are just true today. We know that food safety garners a lot of publicity, partly because of the reach of social media, and also, though, because of better technologies today that allow us to investigate outbreaks. And these include things like pulse field gel electrophoresis, whole genome sequencing, and various collection or database kinds of devices called PulseNet and Genome Tracker, which we'll talk more about. So first, thinking about social media for a minute. And these are data pulled from the internet, just describing what happens in a, quote, 2016 Internet Minute. And these numbers are pretty astounding. If we think about sort of the traditional social media channels today, for example, in 1 minute, it's estimated there are more than 700,000 logins on Facebook, or close to 350,000 tweets. And Instagram, while a bit newer, perhaps social media site, still 38,000 posts to Instagram within just 1 minute. And there's many other sources on here as well. So this indicates that information can travel far and fast. And we need to consider the consequences of, say, food safety news that might be spread through these channels and spread globally. So if we think about food safety in social media, one, it's definitely a good way to exchange information and can often serve as an early warning system of emerging issues. Certainly, as was indicated on the previous slide, the availability of information and data to consumers has grown tremendously over the past 10 years. We also know that consumers and their sphere of influence has also increased as they become more connected with others and informed about things. So we're seeing this new wave of experts who really can alter public opinion. And these folks really do garner a lot of credibility, but we do need to think carefully about how accurate the information is that's being spread. We know that crowdsourcing websites are tracking food safety concerns today, and it's imperative that we start thinking about the role of social media, not just for marketing products and so on, but also in their roles in crisis preparation and in response. There has been some work looking at the question of, can social media actually help investigate foodborne illness outbreaks? There has been some suggestion that people may be more likely to talk about foodborne illness online, sort of anonymously, than actually calling into a health department where they'd have to answer a lot of questions and provide a lot of information. So it's been studied to see if there's a way, by mining social media, to identify so-called point source outbreak, or an outbreak that originates from a particular place, like a restaurant. So CDC published data based on some 2014 work where they actually went in and they mined Yelp and the restaurant reviews looking for references to illness. So they looked at particular symptom kind of words like not feeling well, or diarrhea, or vomiting, or I got sick, those kinds of things. They looked at over 300,000 reviews in this time period, and they did come across 3 what they deemed previously unreported outbreaks that did point to some particular establishments who had some violations. So there certainly is some potential here, but this was a lot of work to go through these several 100,000 reviews to come up with 3 outbreaks. It's not to say it shouldn't be done, but it does take some effort and a lot of resources. I think over time, though, this will improve and really give us some benefits on potentially providing more information to help us with outbreak investigation, and is certainly another tool in the toolbox for disease investigators. But the reality is we really need to think about the flow of food from farm to fork, because there are issues that can arise across this whole continuum. Certainly things back on the farm, through transportation and processing, further distribution, then in a food service venue, and then certainly at the table or in the consumer's hands as well. But the reality is that this global supply chain which we have today is increasingly complex. And here's a graphic pulled together by USDA just highlighting a hamburger type sandwich and the potential sources of all of the ingredients that would be used in it. If we look at something relatively complex like the sauce that might be used on this hamburger, just look at the numbers of countries from which the paprika spice as a minor ingredient in this sauce could come from. Even if we consider something more simple, like the lettuce that might be used on this hamburger, look at the wide range of countries there. And that's the reality of our food supply today. In order to serve people around the world a hamburger, again, a relatively simple kind of food, means we need to pull foods from all over the world. But what do we know about all of these countries and all of the particular points along the chain where food might be handled? There are risks with this, and there was an outbreak recently where foods processed and produced in the state of Washington in the U.S. reached across the Atlantic Ocean and led to recalls in the United Kingdom based on those issues that occurred several thousand miles away. So we hear about the idea to source more locally sourced foods and so on, but that doesn't necessarily mean a less complicated one, or that attention to these basic food safety programs needs to be there as well. So bottom line, in our world today, certainly through social media, but also through other channels, there is increasing numbers of and pieces of data out there. Just think about how quickly information can move that allows us to be aware of these risks and monitor them carefully, and then allow us to respond. Technology has certainly improved. Think about the complexity of your cell phones today versus maybe 10 or 15 years ago. And I think it's important to think about how these tools can help us manage potential issues. But the data alone aren't enough. We need a way to pull insights from these pieces of data, which are tremendous, to allow us to develop actions to solve the problems of the world. But with this increased knowledge comes more responsibility that we must be concerned about. And one of these sources and tools, but also adding complexity, is a technology called whole genome sequencing, or WGS. This is a technology that allows us to identify the complete DNA makeup of an organism, and DNA or genetic material is part of every living thing, including bacteria as well as people and animals. And this variability can help us understand different species and variations within those species and between different kinds of salmonella, for example, or to allow us to distinguish a salmonella cell from a listeria cell. It's much more precise technology than we have had in the past, and is allowing us to identify a pathogen or disease-causing organism that might be in a food or an environment, and comparing those to isolates from someone who is sick. With this identification, we can make a reliable link between, again, the food source and the patient, which helps us to define the foodborne illness scope and where it's occurring. And we're seeing increasingly that this can span over many years. So how about thinking about whole genome sequencing in more practical terms? We certainly know it's more precise, and I use this analogy of a car to illustrate this. With previous technologies, we would get to the level of, for example, knowing that the issue was a blue Chevy. Well, just think about, as you're driving around, how many blue Chevys or blue cars in general you would see. There's a lot of them. With whole genome sequencing, we're allowed essentially to get down to the vehicle identification number, or the VIN. And this is the number that each car has a unique one, and it tells us all kinds of information about that car, where it was built, who manufactured it, its brand, even the engine size, what year it was built, where it was assembled, and then a unique serial number, again, giving us a lot more information than just a blue Chevy. And that's the power that we can get with whole genome sequencing as it relates to the identification of an illness agent. We also know that whole genome sequencing is pretty fast and inexpensive as well. 20 years ago, we generated the first sequences at the cost of about $1 million a piece, and these took more than a year to generate. Today, the technology has evolved such that it costs about $50 per sequence, and that's dropping, and it takes just about a day to do. We also know that the reality of microbiology and the science is evolving. We have improvements in cultural techniques with very specialized systems in order to differentiate organisms that has occurred through the years. And today, with whole genome sequencing, we can use this technology essentially in a one-test-fits-all, where we can use it to hone in on a particular virus identification, maybe a bacteria, a parasite, or a fungi, really regardless of its source, again, from a food, an environment, or even a patient. And here's a couple of examples illustrating how this was used. In 2015, there were Listeria cases attributed to soft cheese. And it was detected, and here's the 2015 data over on the right-hand side of the slide. This represents the cases through the years. And what started this off is, again, in this 2015 time frame, sequences from patients of the Listeria cells, which were isolated from them, were investigated. They did epidemiology, working with these patients to find out what kinds of foods they ate, where they had been dining, et cetera, and that started the investigation. From that, they eventually matched these patient isolates back to soft cheese, which was manufactured at a dairy facility. And then they went back into the database of so-called unsolved or cold cases of illness back in the time frame from 2010, for example, and those isolates from the patients back here also were closely related to the environment. So here they had an outbreak that included 30 cases, 28 hospitalizations, and 3 deaths ultimately, spanning 5 years. And the most cases that were ever detected at one time were 3, or 2, or 1. And in the past, my guess is this outbreak would probably have gone undetected because these low number of cases would never have been big enough to generate a response. Certainly they should, and certainly there are issues that needed to be addressed, but they essentially would be under the radar screen because we just didn't have the technology. Here's another example that occurred more recently related to Listeria, again, in frozen vegetables. And here, these case numbers are 1 over these several years. The outbreak ultimately involved 9 different people over 3 years, and there were 3 deaths. Here we saw that whole genome sequencing was used to link these cases together, and also to vegetables which were produced from one particular processor, which when they went and looked at it more closely, they did isolate the same strain of Listeria monocytogenes from the plant that linked back to the patient isolates. So again, 9 cases over 3 years. In the past, this would have been under the radar and likely not detected. Part of the power of whole genome sequencing comes from the databases into which all of these pieces of information are placed. And one that's in wide use today is called Genome Tracker. And this is a network of laboratories around the world who are using whole genome sequencing to identify pathogens. It allows them to collect and share this genomic information as well as geographic data from foodborne pathogens, which could include patient isolates, as well as data from facilities. So it enables this comparison in real time to help speed outbreak investigations and reduce foodborne illness and deaths. And this graphic down here illustrates, on the left-hand side, starting back in 2013, virtually there were no isolates stored within Genome Tracker. Fast forward to today, first quarter of 2016, there were more than 50,000. And certainly this number has increased over the past several months. So just look at this virtually exponential rise in information in this database. Also, there are at least 8 different countries who are contributing information to this database. So if we think back to our somewhat simple hamburger example, whose ingredients were sourced widely from around the world, think about how we could potentially link information that might be contributed to Genome Tracker from these countries to illnesses that might be occurring in a different one of these countries. So we can look at these data and apply it in a variety of ways. Certainly it can help us to identify harborages or sites where agents might accumulate and might need some attention. It can also tell us about outbreaks. Is the current definition changing? Do we need to change the definition of a case of who's getting sick? And can we really link all of the cases together as we might have in the past? Do we need to split them out, or do we need to combine them together? That can also tell us information about the particular illness agent, the virulence properties of the particular bacteria. Is it resistant to certain antibiotics? And certainly can help to identify emerging food safety concerns attributed to different kinds of foods, to different kinds of agents from different parts of the world. It really just arms us with quite a lot more information. And this technological advance can really improve public health. We have seen that the number of identified outbreaks has increased. And in fact, this quote from the CDC is quite telling. It was stated earlier this year that this collective system, that is with whole genome sequencing, will find more outbreaks, and will find them when they're smaller. That means we can identify what's going on and get in there and take action against it so it doesn't get larger. So we see applications in monitoring of food, in surveying for disease, including identifying sources, identifying attribution or what kinds of food might be implicated, how the agent might be transmitted. So it really helps in our outbreak investigation and response, and can also help assess the effectiveness of various food safety risk management strategies that might be put in place. So all of this is very, very good news, again, from a public health standpoint. But with this comes some other implications. First of all, increased emphasis on this zero tolerance for this so-called human illness standards, because these single cases, as we showed in the couple of outbreaks that I highlighted, they have greater potential to be solved. So increasingly we are seeing a real zero tolerance for any human illness, which certainly should be the case from a public health standpoint. Regulators have these tools in hand, though. So there will be more sophisticated testing, or there is more sophisticated testing, and we may see more strict regulations as a result. Industry has a role as well. We need to enhance prevention efforts. We need to flawlessly execute any preventive controls that would be focused against these risks along the whole farm to fork continuum. We need to think about what kinds of new or innovative risk management solutions would we need to put in place. And then we need to use our bioinformatics expertise, or working with colleagues who have this, to help us manage these data, to pull insights out, to use it to inform decision making. And WHO announced earlier this year that it's quite likely that the Genome Tracker system will be expanded, which means that the numbers of countries inputting data into this system will expand. Now, this is a new reality today. This faster detection of outbreaks does impact real-time business decisions and decisions about what production might be done, for example, from a regulatory and scientific basis related to compliance monitoring, monitoring public health data, also getting into the realm of predictive analytics. So overall, these risk management strategies are becoming more complex. We need to identify what critical items need to be tracked and ensure compliance to basic practices like sanitation. Certainly strong partners across the supply chain can help identify, prevent, and resolve issues before they expand. So how do we respond to all of this? Certainly assessing particular operations and the risks they may present and the foods that are there need to be assessed and action needs to be taken. So this could be in a particular location where you are working, but also thinking along the whole supply chain that feeds into that. Verification needs to be strengthened, and these include activities such as training, inspections, perhaps enhancing various monitoring at appropriate points in the supply chain of in-process product, finished product, maybe the environment. Certainly investing in food safety with the right tools and technology, partnering with others to make sure that you are informed, and implementing approaches where they make sense. So bottom line, it gets down to building connections and being persistent. So that is our call to action, and I hope you will use this information to make some of these decisions within your own operations. I will now turn this back to Miriam, and we do have time for questions.
Well, thank you very much, Ruth. That was really interesting. Whole genome sequencing has been a topic of conversation at many conferences recently, and understanding how that whole detective system is working and how all the dots are connected, I think is really fascinating for us now, but also as we go into the future, and the detection will just get better and better and really help us in terms of our food safety knowledge and how the system is going to work out there. Before we go into the questions, and people can still send in questions using the Q&A function, I wanted to take a moment to let our devoted audience know that I'm retiring in January. After 25 years with EcoSure and working with our webinar series since we started it 6 years ago, it's certainly going to be bittersweet. But I wanted to thank all of you for participating, and hopefully our paths will cross again. And I'm sure that the webinar series will continue. Even without me, none of us are indispensable, but it really has been a pleasure working on this over the years. Now, getting back to the questions, let's see. It seems that I hear much more about food safety these days in the news and popular press. Is our food supply less safe than previously? That seems to be the indication in some of the things I'm reading.
Really good question, and before I answer, I would like to publicly thank Miriam for all of her food safety guidance that she's provided, certainly to me, but also, I suspect, to many of you through her many years at Ecolab. I personally will miss her a great deal and thank her very gratefully for her time here, so thanks. But to the question about the safety of our food supply, I would contend that no, our food supply is very safe, but these incidents of food safety concerns are increasingly much more visible today, and we need to be attuned to this because it certainly gives the appearance of more problems today. I think we're just, again, better able at identifying broad issues, and tools like whole genome sequencing helps us to identify and differentiate these organisms and issues with more precision. Again, it's good news from a public health standpoint in that we will, I think, identify things and problems more quickly, but it does put the responsibility on flawless execution of these programs across the supply chain.
Will whole genome sequencing be a tool that can be used in terms of any predictions of food safety issues that haven't come to light? Will it be part of testing programs, I guess, is the question? Well, that's one of these questions that's probably a maybe kind of answer. It really depends on what would be done with the data. Right now, we have the ability and are honing in on
some of the bigger risks for foodborne illness. So, for example, any patient who presents with a salmonella or an E. coli or a listeria illness, and as well as some others, and it's identified that they have these illnesses, the information about the sequence of those particular organisms is uploaded to these databases like I described. Not to say that other kinds of issues that might not, or other kinds of agents that might not fall into these sort of more common categories might not be put there as well. But again, the focus has been on these top causes of illness. There is, though, potential to use things like whole genome sequencing to get a more general kind of picture of sort of the general categories of organisms that might be within a facility or a food or the environment, essentially to go out, take a sample, and sequence everything and to get a picture of that. And it may not get down to identifying the particular organisms. That's being increasingly discussed. How that may be used, I think, is still a bit up in the air as far as what might be done with that information. But certainly as the potential is there to get, again, a sort of genetic picture of one of these environments and then to enable comparisons over time of, you know, an environment in the winter versus the summer months, an environment in this particular kind of facility that's preparing these kinds of foods to one that's preparing a different kind of food. Through that, we might identify some emerging concerns, but we need to dig in and see. So long answer, but bottom line, I think the best answer is stay tuned, and there's likely potential there for that.
Okay. This next question might be asking the same thing. Can we start using whole genome sequencing to track potential issues from suppliers that have counts on their third-party microbial samples?
Theoretically, yes, although I would not necessarily recommend going out and performing lots and lots of sequence testing without a lot of other information. I think it's very important to put any kind of result within the context that it might be generated and to understand the various contributors to, for example, high counts in a food product, for example. You know, just because a food item or a surface has high counts doesn't necessarily mean there's a bad problem going on. Now, it could, but one needs to really consider all of the factors that can influence a high population in a particular type of sample. Things like, how was the sample handled in between when it was collected and tested? Was there particular kinds of foods in that sample that might inherently have high populations? Things like cultured products like yogurt and cheese or fresh vegetables have high populations of organisms just as a matter of their natural makeup. And high levels of organisms in samples that might contain those kinds of foods would not necessarily indicate any problems, and again, would be just sort of what you would expect. Also, really taking a hard look at the practices and procedures that would be put in place around where that sample was collected. Is there a lot of human handling of that food product, for example? Human beings inherently have a large number of organisms associated often with our skin, for example. Again, they may not be harmful, and in fact, most cases are not because we generally are healthy people. But that can certainly influence those counts. So there's a lot of other considerations that need to go in to thinking about the sample and where it came from before I would jump to doing these kinds of testing.
Thank you. So in other words, there are different types of third-party sampling that's being done in manufacturers and producers, but that's different than whole genome sequencing.
Yes. yes.
Okay.
But we need to keep in mind that if some of these, again, more commonly isolated organisms like a salmonella or a listeria are found through these sampling, whole genome sequence testing is being done on these samples, especially if it were collected by a regulatory agency. And this information is being uploaded into these various databases that I described.
Thank you. And in terms of databases, there's a question here. So does the CDC report to the trackers of whole genome sequencing, or I guess how does that whole genome sequencing group work in terms of sharing information?
Well, the databases are, quote, publicly available, so that someone has the knowledge and expertise to be able to go in and essentially look through these sequences, and they're not something that, kind of the average person could go in and look at, but again, they are available. And there's information being inputted here from health agencies like the CDC or various state health departments that would be related more to the isolates from the patient. So again, someone who might be suffering from salmonella, if that was identified and the sequence run, then that sequence would be uploaded to this database. At the same time, other parts of our regulatory system, such as those agencies who are looking at food plants or in facilities who are collecting samples, if they were to happen to isolate a salmonella, for example, from one of these places, they would sequence that, and that would also be uploaded to the same database. It's the mining of these data and again, that connecting of the dots between what might be in there from a patient against what's in there about other sources like non-patients, or connecting one patient to somebody else, perhaps in a different part of the country or a different part of the world, that the real power comes. So, bottom line, the database is open. It certainly takes some skill and expertise to run these sequences, and then there is a protocol to be followed for uploading the information. But skilled analysts can certainly do this and then start looking for how it connects to other information out there.
Yeah, thank you. So how can I be best prepared to address these types of risks?
Really good question, and there's a lot of them out there, and certainly these are increasing today. First, I think using data like we shared in the early part of this webinar, understanding where the risks are. What are the food sources that are being more linked to food issues today? Are you serving some of those? Are you handling some of those? What kind of operation do you have? Are there some of these behaviors in place that might not be optimal that have been linked in the past to foodborne illness? So focus really on kind of the picture of the potential risks that are there, and then making sure that you have a very good understanding of how you, one, need to manage all of those risks from the food, the environment, perhaps employees, and then make sure those risk management strategies, which is cooking of food and storing of food, proper sanitation of surfaces and utensils, and certainly attention to personnel practices are identified but also implemented and continually implemented. So that gets to essentially being ready for the result that might come, but making sure that you're controlling this through the programs, use of the right products, emphasis on the right behaviors, et cetera, to show that these are being implemented appropriately. And if there are issues detected in some of this implementation, like people aren't washing their hands when they are supposed to, focusing efforts on training people or providing them with better tools that may enable them to better wash their hands at the frequency that, or using the techniques that they're supposed to. I think it's much better to start with the things that are kind of easily identifiable and can be addressed right at hand with the particular situation to really help address the risks that could be presented. Because although in the previous question we talked about how there's more knowledge, more information about food safety out there and potentially more issues being reported, the reality still is that our food supply is pretty safe. So it's not like these issues are necessarily happening all the, or the incidents rather of illness are happening all the time, but there may be behaviors kind of going on that need to be addressed before they result in one of these outbreaks occurring.
Thank you. So with that in mind, should I start swabbing my restaurant as part of my QA program?
I would say not necessarily, and I would not suggest going to that step until you make sure that all of these other things are in place. So again, understanding your restaurant thoroughly and the risks that would be presented by the types of food, the type of environment, the types of employees, the types of practices that are being followed. So really, again, getting a very, very detailed picture of the risks that could be presented. Focus on what's really being done in that particular operation, and are they adhering to the tried and true management practices that should be in place to address those. Making sure the right products are available, the right tools, the right training. Are there audits done? Are there responses taken to any results that would be concerning? Certainly before one would get to the point of swabbing, just as kind of a last result test. We need to keep in mind that a swab result would be a snapshot in time, literally reflective of what was going on when that swab was collected at the particular minute, literally, that it was collected. And things could be very different 5 minutes before or 5 minutes after, depending on what's going on. And it's far better to have a general understanding of that environment before I would recommend, again, just jumping in and swabbing. Finally, related to swabbing, make sure you're ready for the result that you don't want to get. If a particular swab or surface rather is swabbed, and all of a sudden you do identify the presence of a pathogen on that surface, think very carefully about what action you would need to take, one, to protect public health, but also in the immediate terms, but also on what changes that might mean to the particular location. Do foods need to be destroyed? Do people need to be advised? Does a place need to be shut down for cleaning and thorough sanitizing? There likely is a lot of consequences to a positive result, or that, again, that result that you don't expect you'll get. But you got to think worst case, and what would I do if I did find that?
Yeah, well, thank you. And that is a question that we routinely get at EcoSure as part of a food safety program, should we be doing this? And I think that's a great point. What will you do with the information? And often you don't get the results for 28 to 48 hours. So how do you know what really happened 2 days previous?
Yeah.
So
or
it is kind of,
yeah, and sometimes it's longer, depending on how the sample might be collected and shipped off to a lab, et cetera. So days later, what do you do with that?
right. Right, and obviously this is in terms of food service that we're talking about, and not manufacturing, where sampling is part of normal everyday happenings.
But still, there needs to be a consideration, really, in any environment where a sample is collected. Again, what am I going to do with the result? And am I ready to take the sometimes aggressive actions that might be needed if the result came back showing what I didn't want it to show?
Thank you. And then we have a question about norovirus. It is norovirus season over the winter here. If norovirus is the top pathogen of concern, will whole genome sequencing help me, and how can I use that information to prevent outbreaks?
I would say theoretically, whole genome sequencing could be a help, but as we've been talking about for the past several minutes, it's really important to do all of the upfront work and make sure that the right practices are in place, the right behaviors and the right procedures are in place, implemented, and constantly reinforced before I would ever suggest getting to the point of using swabbing. The challenge, too, with norovirus is that it is an organism which mutates or changes its genetic material very quickly, partly in response to its being able to continually survive, and the whole notion of survival of the fittest certainly applies to norovirus. It wants to survive and continue infecting people so that it can continue to live. And with these changes in these genetic parts of norovirus, it means that it theoretically is much more difficult to track this kind of organism if it's changing all the time. So a sequence that might be detected today might be very different than one that might have been theoretically detected a time ago, or even in another part of the country. So we need to think really hard about the biology of the particular organism we might be looking for when we go into this. So part of the reason for this webinar was to not necessarily have you all run out and do lots and lots of swabbing, but to think very carefully about the implications if you chose to do this yourselves, but also of some of this that might be being done out there, and to help you be better armed with information so you're more prepared to deal with these potential issues.
Well, thank you so much, Ruth. I think with that, we've run out of our question time. So I want to thank you all for joining us today. And as usual, there will be a recording of this presentation. It's going to be available on Friday. And if you go to ecolab.com, media/center/presentations, you can find it there. If that's too long to remember, if you just go to ecolab.com and in the search box just put in webinars, it will take you where you need to go. And don't forget, at the end of this broadcast, there is a survey, and this is also where you request your continuing education certificate, so we appreciate your feedback. And then also wanted to let you know that the Global Food Safety Conference will be taking place at the end of February into early March in Houston. And this is where experts from around the world come and talk about food safety issues and network and share knowledge. And this is always a wonderful conference, and so I would encourage participation. And you can get more information under ecolab.com/foodsafety or going to tcgffoodsafety.com. So thank you for joining us this year. Watch your inbox for information on next year's Ecolab Food Safety Matters webinars. And I want to wish you all happy holidays and best of luck in all of your futures as I go on to my next adventure. Thank you all so much.